Hello, everyone. My name is Justin Balko. I'm an associate professor of medicine and pathology, microbiology, and immunology at Vanderbilt University Medical Center and Vanderbilt Cancer Center. Thank you very much to BioNano for having me give this presentation on some of our work together. The title of my presentation today is Optical Genome Mapping of Large Structural Variations in Non-Inflammatory and Inflammatory Breast Cancer. So we, for a while as a lab, have been very focused in breast cancer, and particularly immunology of breast cancer. And for a while now, also interested in this one very rare subtype of breast cancer called inflammatory breast cancer. So inflammatory breast cancer is defined as a clinical subtype, and it has a particularly poor outcome. So it has a very early time to metastasis in patients. It seems to have a disproportionately poor response to chemotherapy, followed by complete mastectomy, or it requires complete mastectomy in many cases. And it's disproportionately hormone receptor negative, and it has high rates in African American patients as well. Although importantly, inflammatory breast cancer can encompass all types of breast cancer, including hormone receptor positive, HER2 positive, and triple negative breast cancers. So the clinical signs that define inflammatory breast cancer are what pathologically we would call T4D, which means metastases to the local region, the skin. Also a feature called peau d'orange, which is an orange skin-like inflammatory effect on the skin near the breast, near the site of the primary tumor, as well as mastitis-like symptoms, which often result in a very difficult diagnosis, or in some cases a misdiagnosis. Pathologically, we define inflammatory breast cancer as having these dermal lymphatic invasions of tightly clustered tumor emboli. And what you'll see here on this H&E stain is that in the dermis of the skin, pathologists will identify these clusters of tumor cells that are migrating out from the primary tumor and invade into the lymphatic system, causing these swelling and the peau d'orange and clinical symptoms that I described previously. So for a long time, researchers have been interested in the genomics of inflammatory breast cancer. There's been several studies on the genomics and transcriptomics that have been performed and reported in the literature. However, many of these studies, due to the rare nature of inflammatory breast cancer, have been heavily confounded by subtype imbalance, small numbers of patients, poor or non-existent false discovery rates for identifying features, and some modest enrichments for pathways, which are already really known to be associated with technical noise or triple negative breast cancer, HER2 positive subtypes. As you recall, inflammatory breast cancer, while it includes all of these subtypes, seems to be biased towards TNBC and HER2 subtypes. There's been a number of different techniques used: RNA sequencing, targeted next generation sequencing, exome sequencing, and most recently whole genome sequencing. The unique and very distinct clinical and pathological features of inflammatory breast cancer suggest that there's an underlying pathognomonic alteration. What I mean by that is some kind of genetic alteration that is really synonymous with this feature of inflammatory breast cancer that really defines this biological subtype. And so we hypothesize that since all of these things have been done, really to no avail or no defining feature, one poorly captured area by these prior techniques that remains to be explored would be large structural genomic variations, which are really quite difficult to detect with any of those other technologies. And so in order to assess this, we utilize the Saphyr platform to test the hypothesis. This is just a data summary, initial set of samples. I know you guys are familiar now with the technology from prior presentations. Our initial set that we ran on the Sapphire instrumentation or pipeline included 2 inflammatory breast cancer cell lines, some 149 and 190 T, an actual clinical IBC sample, 2 comparator triple negative breast cancer primary breast cancers, and then an inflammatory breast cancer patient-derived xenograft model. And so if you look at these summary statistics, you'll see that they're quite similar for the detection, for the number of data molecules collected, and the different types of variants that we identified within each one of these samples. So no huge biases within this initial data set. These are just some of the Circos plots showing the structural variations that we identified. And so what I want to really get to the point on here that's of big interest to us is that we found, of course, many different types of large structural variations in these samples, but most specifically, one of the things that interested us was something that hadn't really been defined in any breast cancer prior, and those were deletions or large structural variations in the OTS2 gene. And those are identified here. And you'll notice that they occurred in one of 2 different IBC cell lines. They occurred in the clinical IBC sample and in the patient-derived xenograft from an inflammatory breast cancer patient as well. We also did identify one ODS2 insertion and duplication in one of our control non-inflammatory triple-negative breast cancers. Now, if we take a little bit closer look at these ODS2 large structural variations we identified, you'll notice that we've outlined where the introns and exons of ODS2 are here in chromosome 7. What was really interesting was that the non-inflammatory breast cancer in which we identified an ODS2 variation in, shown here, was actually in one of the very large intergenic regions. So at least as far as we know, this wouldn't be predicted to alter any kind of gene transcription or function. However, for all of the other IBC samples where we identified ODS2 lesions, each of these included at least one exon. And in many cases, at least in the clinical samples, very large deletions of many exons towards the C-terminus of the gene, or the 3' end of the gene. So just to show you some of the data supporting this, you can see that there's a large number of molecules here that really support the deletion of the N-terminus, the C-terminus of the ODS2 gene. These are reads or molecules that were identified from the PDX model that we incorporated into the analysis. And you can see that the copy number here really does show that there seems to be something going on. So both the copy number analysis as well as the molecule support seems to show that there's a deletion here in the C-terminus. So what is ODS2? Really, ODS2 has not been studied in the context of cancer at all. This is some pictures from what's called ODS2 syndrome. So it's actually been described as being a familial or genetic, I wouldn't say disease, a syndrome rather, that results in low birth weight, feeding difficulties, intellectual disabilities, and microcephaly and mild dysmorphic features. So it's called ODS2 because it was originally thought to be associated with an autism-like syndrome. Other work that's been done in this field has suggested that when looking at exonic and whole gene deletions in large data sets of patients, both control and patients with ODS2-like syndrome, that the greater severity of the disease or the syndrome seems to occur just like what we identified with deletions that are lying in the C-terminus of the gene. And there's an inference from studies that there seems to be a haploinsufficiency without a dominant negative function. What I mean by that is the single copy loss seems to suggest, or where data analysis seems to suggest, that a single copy loss in the germline can cause this syndrome. So this has also been studied in mice, and this seems to, whenever ODS2 is deleted from the germline, in mice, as you can see here, this results in many of the same, at least dysmorphic features that we observe in patients. And this paper here that appeared in Nature a few years ago really helps define ODS2 as being an epigenetic modulator as part of the polycomb repressor complex that can alter this from a repressive to an activating phenotype, the presence of ODS2. So ODS2 is mainly, as I needed to mention there, is really thought and really only studied to be expressed within brain tissue. So the first thing we wanted to do is validate this. We were able to develop, at least specific to the human IBC sample that we analyzed by Sapphire technology, we were able to develop a FISH probe that would cover that region. And as you can see here, looking at the copy ratio, we actually do see what appears to be a single copy loss in about 50% of the cells within the tissue sample by FISH. In a second analysis, we also performed Sapphire pipeline analysis, rare variant pipeline analysis on a series of non-inflammatory cases. We really wanted to make sure that this wasn't a technical artifact, or it wasn't at least something that was very common in breast cancer that hadn't been picked up. So all these are non-inflammatory breast cancer cases. We did 8 different samples. Just really quickly going through the findings here, we accidentally included, I hate to say that, but we accidentally included 2 different regions of the same tumor from the same primary resection that had different sequence ID numbers. So there's a little bit unfortunate, but it gave us a real opportunity to look at reproducibility of the assay going through DNA isolation, different regions of the same tumor, and then also the data analysis. So you can see the high degree, striking degree of similarity between those 2 samples, demonstrating some level of reproducibility. We can also see things we expect. So to some degree, in HER2 amplified tumors, we can see either low or high degrees of amplification, including chomothripsis in the HER2 positive sample over here on the bottom right. And in this secondary analysis, we did identify one more deletion in a triple negative non-inflammatory sample. And this was a very small intronic deletion. So again, not disrupting presumably gene function. And so our conclusions and future directions are that the genomic underpinnings of inflammatory breast cancer still remain to be discovered if they actually do exist. And hopefully elucidation will aid us in not only better treating this aggressive disease, but really understanding the biology behind it. We did identify OS2 deletions that involve the Cterminus domain that seem to track with inflammatory breast cancer status, but we need to be able to validate this in larger sample sizes. And we also would like to determine whether deletion of this specifically in tumor-bearing models, such as the new IRIS-CRE model, would generate any kind of inflammatory breast cancer features. And this would be an incredibly useful genetically engineered mouse model if this is the case. So we're currently in the process of creating floxed mice that can be manipulated to lose ODS2 specifically in breast tumors. So with that, I'd like to acknowledge some of our funding. Many of the things that put this project together, including the bionanogenomics team, our pathologist here, Paula Gonzalez-Erickson, and then a donor of ours, Terry Arnold, who is an inflammatory breast cancer survivor herself, and has really done amazing work in the last 15 to 20 years developing the IBC Network Foundation. We also received funding from Genentech, Roche, and NIH. And with that, I would like to close, and feel free to email me any kind of questions. Thank you.